I'm going to discuss about some attractive repulsive models in collective behavior. So I will give you first the motivation why I was interested in this problem and from where it came, the interest precisely in some of the questions. And then I will come back to the experiments that trigger my attention to this problem later on in the talk. So in fact, as I said, my motivation was to understand a particular experiment that a collaborator of mine, Hideki Murakawa in Japan, showed me a couple of years ago, or yeah, more than that now, 3 years ago, about cell sorting. So let me explain you the experiment that they were doing at the time. Hideru Togashi was his collaborator in biology. And they were growing a certain kind of hepatic cell from mice, and in such a way that when they wanted to see what was the effect when the two fronts of these colonies were meeting, what was the effect of certain genetic manipulation they were doing on some cells. I will explain in a sec. Let's just fix in the first row here. So our attention here, and then you see that when they grow these colonies, and then when they meet in a very nice, sharp way. There is no, there is a sharp front, say, between the two, and not intermingling between the two colonies. And then essentially here, they grow the same kind of cell, even if they were colored differently, they were the same kind of cell. They were not genetically treated or manipulated. And then in the second row, what you have is that on the right, the green one was genetically manipulated. They change one of the pathways for expressing certain proteins that are governing the production of certain cell adhesion molecules. And then in some sense, they are changing the ability of those cells to adhere to others or to the other kind of cell, no? Because I mean, these are the ones that were not the red ones, they were not genetically manipulated. So in that case, what they see is some kind of invasion front or some zone of the intermingling of the two type of cells in the middle. So they were trying to understand if they could explain this based on well, some models. Okay, so another well, more kind of experiment also that took my attention related to the same kind of problems is the pattern formation in zebrafish. So this is quite, in fact, it has been there in math biology for many, many years by now. But I was visiting precisely my collaborators in Japan, and then I met Shigeru Kondo at Osaka University, which is one of the researchers, well-known researchers that have worked in studying these kind of issues, particularly in zebrafish. And well, he explained me a little bit some of the biology behind the formation of the stripes in this particular fish, and that they know very well because they have conducted many, many experiments in his lab. Okay, so let me start with some pictures of my old campus at Imperial College in London to tell you a bit the idea of the kind of model that I want to develop. Okay, so essentially, here you have three views of the main lawn of the campus there. So you can look at it very much at the macroscopic level, and then you can see the real interaction between the individuals at the time in which the social distance was not too strong. And now, I mean, you can, of course, look at it from farther and farther and farther, and then you could see, I mean, you can look at them or try to model them more a kind of population-based dynamics. So essentially, today I'm gonna try to connect from the individual-based models at the microscopic level to some kind of macroscopic level models, trying to explain some of the features that we have seen before. Okay, so let me start with some basic things about the basic bricks of modeling that I'm gonna use. I'm gonna try to think about this in terms of aggregation-diffusion systems. So I will get to this later on in my slides, but let me start by explaining this in basic terms. So let's assume that we are discussing about the interaction of n particles. You can think about them as n cells if you want, and you are modeling them just by looking at the nuclei of those cells. And you're going to assume that the interaction is due to certain forces. And I will explain what's the origin of the forces that I'm thinking for the application of the cells in a slide later on. But right now, let's think even about in terms of physical point particles. So I'm going to assume that the presence of a particle at position xj is going to produce a force on xi, which depends on certain potential. And the potential is going to just determine the force by taking the gradient. And then is directed through the line joining the 2 points, and then is given by that formula here, gradient wxj minus xi. So here what I'm writing for the evolution of the particle's position is nothing else than assuming that the inertia term in second order equation is in a Newton's-like equation is negligible, and assuming that we have some damping, and then I can just say that directly the velocity of each of the particles is equal to the sum of the forces. And here I'm taking a particular scaling to be able to say something about the system when n goes to infinity. So this 1 over n here is already important because I would like to see what happens when I have a large number of individuals, like it would be in practice if I want to use this for describing a large set of cells. So just for notation, normally associated to a particle system, you have what you define as the empirical measure, which means that I put a Dirac delta at each of the positions of the dynamical system at time t, and I normalize it in such a way that it's a probability density by the factor 1 over n. And just to keep an idea about the shape of the potential, typically I'm going to think about potentials which are repulsive for short distances and attractive for long distances. The short repulsion makes sense, as I will try to convince you in next slide, in a particular way in these kind of applications. Okay, so just to go quickly over this basic model. So if you go to the limit somehow n going to infinity, it's not too difficult in the case in which W is good smooth potentials to see that what you expect as a kind of PD model at the level of the probability density or the evolution of the mass density, depending if you normalize or not, is this kind of continuity equation with that velocity field which is obtained in terms of the empirical measure by doing the convolution with the gradient of the potential. So, and the interesting thing about that is that already this basic model gives you a lot of, there is a lot of variety in the possible steady states and the shape of the solutions, as you will see, depending on the particular properties of the W. So after this, let's go back to the real objective. So you want to use this for the modeling for this population of cells. Let's try to make it more realistic, the model, introducing some, at least some basic effects. So the first thing I want to include is the type of interactions. So let's assume that, in fact, the repulsion is very strong if the distance between the nuclei of the cells is small, smaller than certain value, certain distance, which in fact is twice the size of the nuclei, the typical size of the nuclei. So this is what I'm going to take as a kind of cutoff. So I'm going to assume that if they are very close, there is a very strong repulsion. And this is just because of incompressibility assumptions. So they occupy some volume. And then the volume that they occupy, that's why I'm saying that there will be a very strong localized repulsion. Then I'm going to assume that there is a, if they are between certain threshold in the distance between the two, that they have some tendency to adhere to the other. So there is some adhesion forces, which typically in many of these experiments, and in many of the, in particular in these two applications I show you, most of these adhesions forces, you see then that they are produced at a longer typical distance, at a longer typical scale of distance, because they are produced by, let's say, long philopodia, long protrusions that the cells do in such a way that they connect with others. We will see that in some of the pictures later, in some of the experiments. And then, of course, at a certain distance, there will be no interaction, okay, to make it realistic. And of course, if I want to do something also for some of these experiments, what I have to consider is different type of cells. Because we discuss in those experiments, either for the pattern formation or for the cell sorting problem, that typically you will have different type of cells. And then the basic way of introducing this is that I'm going to assume that at least I have kind of two species model. And two species model in the sense that I will have two type of cells, if you want. And then I can assume that the different potentials of interaction between the different type of cells may be different with different strengths, or even different shapes, depending on the kind of model you want to make. Yeah, it's not difficult again to check that the macroscopic model you expect for the evolution of the two probability densities now of each of the two type of cells will be a system of that form, in which now all the forces that you have here, they correspond to the interaction between the first type of cell and themselves, the first one with respect to the second, or the second with respect to the first, and the second between themselves. And of course, the W12 and the W21, and that's the most important point to make in this slide, is that they don't have to be symmetric. Of course, the red can like the green, but the green maybe doesn't like the red, and they try to escape. Well, it's just a way of picturing the interaction between the two. So this doesn't have to be symmetric at all. I mean, it depends on the kind of model you want to make. So another effect that I want to include at this level is the incompressibility, say, of assumption I made before. So this hypothesis, I said that I want to include the fact that these cells occupy a certain volume. And how I can do that? By doing a very strong localized repulsion at zero. So in fact, out of all of these potentials, I'm going to assume that there is a very strong localized repulsion at zero. What it means that I'm going to model that by putting a Dirac at zero in the potential with certain strength in each of them. So in fact, I'm assuming that each of them has that part. And by getting rid of the tildes here and putting the Diracs inside, what you get is the last term that I'm writing here. Because if you put a Dirac at the level of the potential, of course, this gradient is not going to be on the potential, but rather on the density. So then you will get typically the gradient of the density times the density. Here I'm changing a bit notation just to think that we are thinking now in terms of functions here, and that we have a term, the term that comes from the Diracs in each of these four terms is summarized here, and it's like for the first species, like rho gradient of the sum of the two species. So it gives you a term which can be interpreted as population pressure. So the total population produces a kind of pressure that brings the densities to spread. And you see that now you have a term also, I mean, you have a self-diffusion in a sense, because this term will give you typically rho gradient rho. So it's a kind of nonlinear diffusion for rho. But also you have a kind of cross term here between rho and eta with the other species. Okay, so let's see this simple model, how complicated it can be. So the first thing I want to analyze is what happens in one dimension. So even in one dimension, I want to convince you that it can be very complicated. And the fact is the following. Let's look at the steady states. Let's just look at the steady state in particular potentials. So I'm going to consider two types of interactions. Either they are attractive. So what I mean is that both of them, W12 and W21, they want to attract each other, the two species. So they are functions which are non-decreasing as functions of the radius, of the distance. And the other case, I'm going to call it attractive-repulsive, in which the first species likes the second, but not vice versa. And then one of them is decreasing and the other one is increasing. And I'm going to take a particular case, because in this particular case, the computations are even, you can do some explicit computations. So for the attractive parts of each of the two species, I'm going to assume that you have a potential which is x squared over 2. They attract very strongly to each other. And for the cross interaction, I'm going to assume that is given by modulus of x. Either positive or negative means if it's positive, it's attractive, negative is repulsive. And why modulus of x in one dimension? Because it's related to the fundamental solution of the second derivative operator. And then it gives you certain particular expressions for the steady states, which make things even explicitly computable in some sense. So let's assume that we get either of these two, and let's look for the steady states. The steady states should satisfy that the velocity field here is 0 wherever you are on the support of the density. And then from there, what you get is that this quantity, this whole quantity, should be constant wherever rho is present. The same for this quantity wherever the eta is present. So that's the relation that you have. So these things should be constant in each of the supports of the two densities. And then you can, I mean, in this case, because of the expressions of the potentials, you can compute. And you can really see that whenever rho is present and the other species not, or vice versa, you can write explicitly in terms of certain constants the solutions. They have to be like, I mean, just kind of parabola. While if both of them are present, you can compute what they have to be, and then they are a combination of these sine and cosines, with, again, certain constants to be determined. So one typical solution that you can find is this one here, which is a kind of, one of the species is around the other one. So it's you get that the blue species is in fact somehow getting around the red species or the red type of cells. And here is, you have something similar. You have the red here, then around the blue, and then again the red outside. So, but the interesting things is that, and the complicated part of it is that you have that typically the steady state will be compactly supported, which is good from the point of view of the modeling. And also the second interesting thing is that you have sharp fronts when they meet. So for instance, in this case of the both of them attractive, you have this kind of sharp interface when from the support of the one of the density to the other. And in fact, you can compute them explicitly, as I said. If you do some assumption about where the supports should be, and then you assume that the masses of the two are given, and you assume that the sum of the densities is a nice function that is continuous, then you can find this kind of profile that, for obvious reasons, we call the Batman profile, because of the shape, obviously. And the interesting thing is that you can, I mean, you see them also in simulations. It's not that it's a stable state. Let me show you one simulation of the system. So if you get a symmetric initial data, you are typically going to have this kind of behavior towards a steady state. And then you see that the solution, even if it's nice and even smooth at the beginning, then it will develop these fronts as t goes to infinity. And they are really discontinuities here. So let me now show you another case, which is the case of attractive-repulsive. So if you have attractive-repulsive, typically you have this kind of behavior also of engulfment as before, but now the interesting thing is that the supports are totally segregated. While before one could be on top of the other, so you have this engulfment but coexistence wherever the other density was present, here they are totally segregated. And you have sharp fronts between the two different type of, the two different species here. And depending on the parameters, of course, you can have something completely segregated. These things, you see, I don't want to discuss much more there, but let me just mention, just to tell you how complicated it could be even in one dimension. You can have, depending on the distribution of the mass of the initial data, traveling waves instead of steady states, but they get the same, I mean, the shape of the traveling wave has the same kind of complexity. So you can classify them between, let me show you here, that's a traveling wave, which has this strict segregation between the two. By the way, I'm not saying anything about the numerical schemes here.
One question.
Yeah.
So then, if we are speaking on traveling wave, you are thinking we don't have boundary condition, right? So
here I'm just for the moment. I'm doing, you want in this case, I'm doing just either no boundary conditions or periodic boundary conditions.
Mm-hmm.
Yes, yes. Nevertheless, in the cases of the stationary states, since they are completely supported, the boundary condition doesn't play any role. There will be also solutions with no flux at the boundary.
So the interesting enough, if we add these two equations, which you show that, then we obtain for the sum a porous medium type equation, right?
That's right, but there is but they are coupled. There you cannot you don't obtain directly the porous medium equation. I'll come back to that in a second. In fact, yeah. In fact, let me just mention in this simulation. Sorry, this one. That one is the case of attractive-repulsive, and you see that even if the initial data they are mixed, then they separate asymptotically. They segregate. The interesting thing is precisely related to what Masoud was just asking me. Let me see if I can get to that, or I do it in the, yeah, okay. Let me see if I can get to the equations quickly. Equations are a bit far back. Here. Yeah. So the thing is that if you add them, this term here, the term of the population pressure will give you the kind of porous medium term with exponent 2 for the sum. But these are fully coupled and it's not a close, you don't have a close equation for the sum. So you cannot write it really in an easy way, alternative way. Only in one dimension there is something that you can do, and we took advantage of that, as I will mention a bit more later. But yeah, that's what is behind. But the drifts are destroying that, that effect. So that's why for the sum you expect better regularity. Sorry, yeah, okay. So yeah, in fact, that's exactly the point that Masoud was asking me. So if you have the equations and no drift, these equations, you can add them, and then you have an equation for the sum, which is exactly nonlinear diffusion with exponent 2. And it's well known that in that case, you have also, in fact, already this behavior present. So you have that when they meet, in fact, if they are originally separated, they don't mix, and you have a sharp front. By the way, this is due to the boundary condition, and in this case, is no flux. So that's why it goes to constant over there.
And the sum will be continuous. The sum
yes, the sum is always continuous. You see, that's the point. But nevertheless, each of the two, they develop a discontinuity. Mm-hmm. That's the interesting thing about this. Okay, so I wanted to mention that, of course, in practice, you want to include also reaction terms. Typically like Fisher-like terms. And this is, in fact, just I'm gonna make several comments about the more theoretical side of it. What do we know about well-posedness of this kind of system before I get to the practical applications again. So, but just some comments about this. If we have reaction terms and no drifts, for that, we do have a reasonable theory already developed, but very recently. So one of the things we did with my PhD student, Marcus Smithton, was to look at this problem with certain assumptions on the red functions over there. It doesn't really matter to get into the technical details here. And the interesting thing about that is that in one dimension, then you can take advantage of the sum, and because you can write equivalently a system for the sum and for the mass ratio of one of the populations. And then over there, you can work and get a good theory that gives you BB bounds, which in the technical is a technical way of dealing with the sharp fronts. Okay, but I don't want to discuss much more than that on the theory. Okay, the other comment I want to make is that we don't know how to do a theory like that, even in one dimension, when you have the non-local terms. With the non-local drifts, we don't have such a nice system. I mean, we cannot write a nice system for the mass ratio and the sum, even in one dimension, and we don't have even a good theory for that in 1D. So let me now skip this one. Just to show you again a numerical simulation of this system, but with reaction terms. The reaction terms were like rho times 1 minus rho, eta times 1 minus eta. Nothing too complicated. Just to show you that, again, this kind of segregation and sharp fronts separation between the two is there and is present. And these are just like snapshots of this evolution here. Okay, so in the last part of my talk, what I want to go back to the original application. So how can I use these equations? So what I did here was try to mimic the first experiment of cell sorting. So let me explain a bit the choice of the different kernels. So what we did in this first simulation is, for the time being, let's not put any reaction term. Let's consider the total population pressure. So this kind of nonlinear diffusion term that gives you the strong repulsion at 0. That models the fact that the cells occupy certain volume. And then let's put only an attractive potential between green and green, green, red, red, green, and red and red, all of them of the same shape. The potential is the easiest potential you can think of. It's just linear, cut off at a certain distance. Okay, so what I mean is that, in fact, what you are doing to compute forces is just an average of the forces in a disk around each point. And then what we did is, let's start with the case in which the strengths of the 4 potentials are the same, so it's essentially the same population. And then let's take the parameter that controls the attraction between red and green and change it slightly. And then the interesting thing is that you start, if they are the same, you start with something that essentially the 2 species are like the same, so they somehow mixed. While when you start changing the parameter, then you go to this typical behavior in which one species goes around the other one, and which is what we refer as engulfment. Then you go to partial engulfment, and finally, well, they start separating more and more, and finally they are totally separated. So in this kind of model, just the interaction, the strength of the attraction between the 2 species is enough to give you this kind of behavior from mixing to complete sorting of the 2 types of cells. And this somehow the interesting thing is that it's a continuous expression of what you see at the particle level. Let me show you some more complicated simulations. So let's, I know that this slide is, has a lot of information. So let's fix ideas first here. In these 4 simulations, what I did is essentially to repeat the same numerical experiment with the 2 species, but with an initial data which was random. Let me see if I can restart it in an easy way. Okay, no, I had to do it one by one. Okay, so the same initial data for the 4 of them. Okay, but random distribution initially. And then you see again the same effect. You go from mixing to some kind of engulfment, and then partial engulfment, finally to complete sorting in the sense of complete separation. And again, the idea was just to touch this kind of the relative strength between the inter-particle, say, attraction with the one in between the 2 only, to go from one to the other. And the work we did also with a group of biologists with, in Japan, Sato, Togashi, and Trash. What we did, it was to try to reproduce the experiments that they, try to reproduce the experiments that they did in their previous work, to try to estimate the length of the interpenetration of between the 2 species, and see if by taking a good choice of the parameters, we were able to reproduce some of them, and eventually even see if we were able to get other more complicated experiments. So the thing here, what you have is the experiment, the real experiment, here in these 2, while here you see you have the simulation of our our model, in which we have already chosen well the parameters. And in fact, we fixed many parameters from just the information coming from the biologist, and we were able just to reduce it to the strengths between the different species in order to somehow to qualitatively get this kind of solutions. Let me show you here, I want to make more comments on the model. For getting this, we had to introduce several additions into the model. One of the additions that we had to do is to put a cap on the total density. So there is this term that was not in my original model. So this 1 minus U minus V. This allows to show that in fact U plus V, which are the 2 populations down here, they cannot go over 1, because we are thinking more in volume fraction of the 2 species. Okay, these K1 and K2 are the same as before, the sum of the forces. So they have all the potentials, the W11, the W12, and vice versa. It's just a way of making a shortcut notation here. Okay. So we then with the yeah, okay, yes, this one is the one I wanted to show you. I will run it again. Once we fit somehow we obtain the right parameters, we use it for other experiments they develop, and then we could also recover other behaviors very similar to the experiments, like this one, in which you see in the 3rd one, which is new with respect to the previous, then the red keeps the sharp front with respect to the green and then pushes back the green somehow. While here there is this pushback of the green somehow the red is pushing back the green, but at the same time there is this interpenetration between the species. So here the simulation is a 1D simulation. We are trying to do the cut of like the kind of perpendicular cut to the front that we see in the experiments. And somehow we were quite happy that the model based on the obtained from the basic interactions was enough to give you this. Then let me now go back to the also to the application for zebrafish patterning. Just give me a sec. Some noise from the neighbors. So for the zebrafish, so as I said before here, the model is necessarily much more complicated because there are plenty of different cells involved that the biologists and these groups of biologists know very well because they have done hundreds of experiments by now on this particular issue. So again, the idea that we develop is similar. We try to write first a model in terms of kind of particles that have the minimal information coming from the modeling. And let me tell you that for the biologist, the minimal model that makes sense is to consider three kind of type of cells. The xanthophores, which are the ones that gives you the yellowish orange color in the stripes, the melanophores that gives you the black bluish color, and the iridophores, which are the silverish type of color that you see typically in the skin of a fish. So again, we use the same kind of idea for designing some of the interaction potentials, and the very basic minimal model that we came with my collaborators is of the kind that I showed you before, in which you have the xanthophores and the melanophores. Somehow we started with the idea that the xanthophores like the melanophores, but not vice versa. And you see here is one of the experiments by Kondo. Here you see the xanthophore, here you see the melanophore, and you see how the xanthophore is trying to catch the melanophore trying to avoid the xanthophore. Okay, and you see also the interesting thing about that most of the interactions between the cells are along these long protrusions that they produce. And you see also the melanophores is trying to catch another melanophore over here and try to get, to anchor over there. So anyhow, so, and this is a picture about the melanophores in the zebrafish skin that has, you see again, the features of the non-locality with these long protrusions they produce. So with this very basic model, in which here the iridophores, we just assume that they are diffusing from the dorsal spine of the fish. Then I'm going to show you some of the simulations. So here, think about this as one side of the fish. So these are the lateral lines, and this is the kind of the dorsal spine of the fish. It's known that this is from where these cells are produced, both the xanthophores and the melanophores. So that's why we assume that initially the distribution of the melanophores is like that. For the xanthophores, we assume that it's homogeneous, and also for the iridophores, they are produced from the dorsal spine. So we just follow, for the initial data, the basic comments that our biologists told us about this. And here is a vertical cut, in which I show you, it's a vertical cut over here, showing you the densities, both yellow and black here. So this is the simulation you get. And the interesting things are, here there is no right-hand side. Again, only the population pressure plus the non-local interaction. And then the interesting things is that you have these sharp fronts between the species produced and the segregation between the species. That leads you to a very clear formation of some stripes patterns, if you want, on the domain. We are trying to improve this model at the moment much more. we develop a more complicated model to try to make it more realistic, following again a lot of the information that is known about the interaction of the different type of cells involved in the formation of the involved in the pattern formation. Here we collaborated with Alexandria Volkenin that has been developing agent-based models for this. And then we follow a lot of the, the knowledge, as I said before, that the biologists told us about the interaction of different cells. So we came up with a more complicated model in which we have either 5 type of cells or 3 type of cells, but kind of more detail, and also in which the right-hand sides they play a role, of course, because there is birth and death of the cells in the tissue, in the growing tissue. And the interesting thing is that we had to include also some non-local terms to make it more realistic based on the information they gave us. Also on the right-hand side, this is something that we are trying to understand better at the moment, even in simplified models. The point of showing you this very complicated model is just to show you the kind of things that then you can get. So repeating more or less the same kind of interaction that I showed you before, the interesting thing now is that you see the formation of those patterns out of the center in a kind of periodic way at the beginning, which is which in normal fish, in normal zebrafish, this is what they see typically in the growth, during the growth. So somehow for them, for our collaborators, this model was, reproduces better what they expect from the formation of the patterns in the growth of the fish. But there are plenty of things here to be improved and also to be understood in terms of the math behind this system that I have over here. And I think with that, I'm almost done. Let me just conclude. And I have a couple of things to say for the people in the audience that are more interested in the applied math side of these systems. Still, we are far from understanding many of these systems, even in one dimension, because we don't know even how to control when you have these sharp fronts, which means to get these BB bounds, which I know that it sounds a bit technical. That's one of the issues. We are able to do that for the case of reaction diffusions, but not with the drifts. So that's still an open problem. And as I said in the last part of my talk, we are very happy that we could get this self-sorting phenomena with this basic model, but still there are plenty of things to understand how to use these for more real experiments. And we are trying to do that and advance that in the research with the team of Kondo and Shekhar and Kataraman in Sussex. And thank you very much for your attention. I hope you enjoyed it.
Okay, thank you, Jose. That is really nice. So I'm wondering if anybody have any question?
So I have, if.
Okay.
So Jose, one of the co-author, Mimura.
Yes, sure. Yes.
In the past, they had such a kind, in your case, segregation results, and I guess even for the arbitrary dimension. What is the difficulty in your cases?
Yeah. So the thing in this particular problem, the segregation result that they were able to show. I mean, in fact, they were not able to show segregation really in this model.
In some sense, they have, if they have.
Yeah, the thing is, I mean, there is a paper of Birch.
Yeah, exactly.
And there is a third author that I forgot. But in that one, what they prove is that in fact, the initial data, they are positive everywhere. They cannot touch zero. And then what they prove is that in that case, they can, they show that asymptotically, they will go to something that is segregated. What we improve over that is that, in fact, our initial data can touch zero. So, I mean, I can have an initial data completely segregated from the beginning, and they will remain completely segregated.
But arbitrary dimension, I guess.
No, this is in one dimension. This is one dimension. In several dimensions, there are no results. There are no results in several dimensions. The result of Mimura is also in one dimension for this. And the question is about the drifts. The non-local drifts is what makes it really complicated.
Later, I can imagine. And then the main idea was, so that if you add this equation, even for simple cases, so in some cases, this system could be gradient system in some particular cases.
In very particular cases, that's right.
And then you can obtain some kind of the existence of solution and something.
Yeah, but generically, they are not.
It's not the case. I agree.
So and that's the real difficulty here, to understand things about asymptotics. Yes. So you can see then even at the particle level, generically the system of particles is not going to be a gradient system.
Yeah, I agree. And when I was young, the Kolmogorov, said it will be interesting to consider, you already started, to consider not the standard distribution sense as the solution as a measure, which you started. So then if you are considering the probability density, you have already in some sense the L infinity estimates for the measure, and it could be are you familiar with this kind of activity?
Yeah, well, that is more in the. I didn't discuss too much on this, but the idea is that you can use that technology to derive the model. You can you can use it for the mean field, what is called the mean field limit. So to get the equations from the particles in a more rigorous way. So I didn't discuss too much on the mean field limit, but this is also a very interesting question, because I scale from the very beginning to get the mean field equations. I scale from the very beginning by 1 over n.
Yeah, exactly.
And this looks naive, but it's crucial. If you don't do that, you are not going to be able to get the continuum equation. And an interesting question would be how, I mean, this assumption, of course, in practice, if you want to use it, I mean, how accurate is the mean field limit for describing a system? So that's an interesting question on itself. Yeah, and how really the agent-based model is in the end going to infinity is giving you a good description. By now, qualitatively, yes, but quantitatively is a hard question. Yeah.
So last time when we meet each other, it was in Oberwolfach.
Yeah, yes, and years ago. Yeah, a couple of years ago.
Yeah,
that's right.
It was nice to see you again.
Thank you.
All right, any other questions? If not, I'd like to thank José for the lecture. Really appreciate that you talking to us about your project. And I guess I'll see everybody next Friday.
Okay. Bye. Thank you very much for the invitation.